Hi, my name is Elena Gofas, and today I'm presenting our work in vivo characterization of a retinal cellular biomarker of inflammation in multiple sclerosis. Multiple sclerosis is an autoimmune inflammatory disease of the central nervous system. And although immune cells play a central role in this pathology, directly imaging immune activity in the brain remains challenging due to its inaccessibility by conventional optical imaging techniques. On the other hand, the retina is an extension of the brain and offers a unique, non-invasive access of the neurovascular network. In the literature, it has been found perivascular immune cells in the retinal ganglion cell layer in multiple sclerosis patients. And so this raises the question if the retina can be a window to immune changes in multiple sclerosis. And so our goal in this study was to see if we could image these retinal immune changes directly in patients. To do so, we use a camera called Adaptive Optic Scanning Laser Ophthalmoscope, or AOSLO. This camera is an in vivo microscope for the retina that can be used directly on patients and offers with a cellular resolution images of the retina, of the different layers of the retina, different type of cells. In this study, we are particularly interested in the ganglion cell layer, where ganglion cells are revealed, but also other type of cells such as microglial cells using this camera. In addition to the microscope for patients, we also perform complementary tests such as visual examination, OCT, or MRI. And our patient cohort was divided in 4 groups: relapse-remitting multiple sclerosis with and without optic neuritis, and also progressive multiple sclerosis without optic neuritis, and healthy controls. So the key finding of our study was that we could observe, indeed, for the first time on patients, cellular infiltrate in the inner retina, particularly in the ganglion cell layer. These cells, although with a similar morphology, displayed different size ranges from 5 microns to around 20 microns. And in some cases, they display one intracellular feature, but sometimes multiples, potentially nuclei. And then the position of these intracellular features can vary inside of the cytoplasm. But the most striking feature of the cells was their strong interaction with the vascular features, particularly in cases we could see these cells partially inside of the vessel and partially inside of the retinal tissue. This suggested cellular infiltration of these cells of this layer. Also, some of the cells with this compact cytoplasm and then an intracellular features resemble a lot of, in the literature, the composition and distribution of lymphocyte B. And in some cases, we observe an interaction between two of these cells that resemble a lot antigen presentation cell signaling typical of lymphocytes. Also, the cells could be followed and tracked over time. Here we can see the case of a few minutes, the cells seem to remain in place. In the case of a few hours, we can see a cell moving above or inside a vessel. And finally, when looking at longer spans of time, we can see the density changing over time. When analyzing this type of tracking videos for each patient, particularly with patients after an acute optic neuritis, we can see that the highest density occur right after the acute episode, decreasing then exponentially over time. But looking at the fellow eye, we can see a lower but stable cell density. An interesting case that we had is this one where a fellow eye presented a rise in cell density at a moment where there were no clinical signs of optic neuritis. However, a few weeks later, an optic neuritis was diagnosed. This suggests that we might be able to see cellular changes before an acute optic neuritis episode symptoms appear. Finally, we didn't find any correlation between cell density and clinical symptoms, particularly optic neuritis features such as length or the presence of intraorbital lesion. In conclusion, Phase contrast AOSLO enables real-time in vivo visualization of inflammatory cells in the ganglion cell layer in multiple sclerosis, but also temporal changes in cell density and unique cellular features that can be detected demonstrate its value for the non-invasive monitoring of inflammation of patients with MS. And so this technique shows promise as a biomarker for MS, but also for other neuroinflammatory conditions. I would like to acknowledge all the people that work in this paper from the different institutes, but also the funding bodies that made it possible.